Hello, everyone, and thank you very much for joining my talk. I am Carlos Outeiral, and I am a last year PhD student at the Oxford Protein Informatics Group in the United Kingdom. I am working under Professor Charlotte Ding on the intersection of protein structure and dynamics, and in particular in protein folding. And today I'm going to be telling you about some results that I obtained in my latest project that tries to understand exactly what is the relationship between protein structure prediction methods and the folding pathways. This project was motivated by some very recent advances in the field of protein structure prediction, which I don't think I need to introduce, but I'm going to do anyway. In December last year, in Cas14, the Google DeepMind introduced a protein structure prediction method called AlphaFold 2 that massively outperformed every other member. In fact, this protein structure prediction method was so good that in some cases it actually outperformed, kind of, the experiments used to obtain protein structures. For example, by identifying the conformations of proteins that had not been resolved properly from the experimental data. This made me think, actually, well, first of all, what are the important questions that are there for poor PhD students like me to think about and to try to solve? And then motivated me to think something a little bit more deep about how we predict protein structures and how we understand the entire process of protein structure and dynamics. And I asked myself one big question, which was, is the success of this method based on um, well, actually understanding folding, actually answering all of these questions about protein folding that we have been asking for 60 years? Or is it simply because we have learned statistical relations about the structure that somehow are simply really powerful, but do not relate at all to the physics of protein structure and to the process of folding? And obviously, we do not have access to AlphaFold 2 yet, or at least we don't have access to it at the time that I'm recording this talk. But there are a lot of methods that are relatively similar that we can look into and try to understand what they are doing when they search the conformational space of the protein and perhaps compare that against experimental folding data. I'm going to tell you about how I collected reference data to try to compare it to simulated trajectories. I'm going to tell you how I generated these trajectories, and I'm going to tell you the conclusions that I got from these trajectories. But before I do that, I think it will be particularly helpful if I show you how one of these trajectories looks like. In particular, this is a trajectory obtained from EBFold, which is a program developed by the Deborah Marks lab at Harvard, in which I simply modified the search engine for the program, which is based on CNS, and made it output a frame every couple of iterations in order to monitor the progress of the folding trajectory. And what we see is something like this. Initially, the protein is fully extended, um, simply completely elongated. And we see how very quickly it starts to collapse and gain some form. It adopts some secondary structure, and then starts doing all sorts of complex movement, really search around it. So you can quickly see that this is actually not very much like what we imagine as folding. It's if I was running a molecular dynamic simulation instead of a optimization process, I would expect to see something much different, where actually I see secondary structure forming first and perhaps then collapsing, and maybe I can see more sort of molten global forms and then formation of strong stable contacts. And it's not exactly what I see, and this is something that is going to be sort of important throughout the talk. But it actually tells me something important, which is that sort of very big changes in the protein structure are going to be what actually drives the folding in these trajectories. So now I have explained this, I'm going to actually tell you how I generated these trajectories. So first of all, I collected from the literature a very large data set of protein kinetics experiments. I say large, but it's actually about 170 protein experiments collected from several databases and a bunch of individual literature points. For this data, I have available kinetics, so I know how the protein folds more or less, the sort of pathway of the sequence of intermediates that leads to the formation of the folded protein. And in some cases, I also have reliable structural characterization by hydrogen deuterium exchange experiments. So I know how these intermediates look like structurally and which parts of the protein are formed and which parts aren't. Then I took 6 state-of-the-art protein structure prediction codes. We'll show them later, but these are TR Rosetta, RaptorX, DMP Fold, EV Fold, which I just showed, SAME2, which is our lab's in-house code, and the classic Rosetta. I modified these codes in order to print the search trajectory as they perform the protein structure prediction. And I ran a large number of decoys for each of the 170 proteins in my dataset. This is relatively simple. In most of the cases, the programs come with bash scripts that do the entire pipeline for me. So I just generated this, ran it through my modified codes, and generated a large number of trajectories. Now, based on the trajectory that I showed before, it's actually quite complicated to extract general patterns. So what I did is try to simplify it as much as possible. I looked at the formation of secondary structural elements, and I looked at the fraction of native contacts between different secondary structural elements. In other words, I look at the native structure, I identify which secondary structural elements are in contact, count those contacts, and see how many of those contacts are generated at every step of the trajectory. And I obtain curves like this. Then, in order to analyze those curves, I simply look at the parts of the trajectory where I see maximum changes. When does a protein collapse? When does a particular interaction between two two secondary structural elements is formed? I cluster those interactions and I identify phases of folding. If I observe that first two alpha helices get together, form a somewhat collapsed structure, and then later two beta sheets get together, then those are two separate intermediates, two separate metastable structures, and I can identify that my protein is folding first by forming this intermediate with these two alpha helices, and then it's forming a different structure, potentially an intermediate or potentially a native structure by adding the rest. Now, using all of this data, I tried to compare it against experimental information. The first and simplest thing that I could think of was to actually compare the predictive folding pathways. But since there are many different possibilities, I tried to simplify it to the maximum and consider only two cases in a binary classification problem. I wanted to determine whether a particular protein will fold via a two-state mechanism, so exhibiting no intermediates, or in a multi-state mechanism where there's at least one on-pathway intermediate. And then I used the six protein structure prediction methods, and I compared how good they are at predicting this dynamics. The results are summarized in this table, which I comment on a lot more in the paper. But just to give you a quick overview, I think the really important results are the majority of methods are pretty bad. I think that would not be an overstatement. If you try to run a statistical test against a random classifier, you will observe that the majority of methods are significantly better than a random classifier, which means that there is some signal in this problem. But the really interesting stuff is if you use a classifier that is based exclusively on the length of the protein chain, meaning you count the number of amino acids in your chain, train a logistic regression on it, and see how well it performs, you will see that actually the classifier based on length is significantly better than any other method. And none of the methods, with all of their information that they have in there, multiple sequence alignments, fragment libraries, lots of parameters from training and other things, they are not able to outperform a classifier based exclusively on the length of the chain, which is also sequence agnostic feature. It doesn't know anything about the amino acids that are interacting there. So another feature that I don't want to comment on a lot in this talk, but that I talk about more in the paper is it seems there's a somewhat of a trend that methods that use more physics, like Rosetta or Saint2, that do not use deep learning but just try to minimize a sort of reasonable potential using Monte Carlo methods, seem to be slightly better, although there is a sort of small sample effect since Rosetta and Saint2 are quite energy consuming, and I couldn't generate a lot of decoys for those methods. The second test that I tried to run is related to the folding rate constant. And this was inspired by a classical paper by Plaxco, Simons, and Baker that noticed there was a significant correlation between the average contact order of the native structure of a protein and the folding rate constant of the same protein, assuming that it folded by a two-state mechanism. So I looked at all of my trajectories and tried to examine whether there is a similar correlation between the trajectories and the folding rate constant. In this plot, I show the correlation between the average relative position of the folding event and the natural logarithm of the folding rate constant. I show this for the 6 methods that I have looked at using the maximum amount of decoys that I had available, and also 2 reference metrics, the length of the chain and the average contact order. First of all, you can see that the results by Plaxco, Simons, and Baker still hold. So the length is clearly the best correlated metric. So sorry, the contact order is one of the it's very highly correlated. It has a very clear correlation. But if you look at the length of the protein chain, it's actually slightly better than the average contact order. But what is really important is if you look at the simulated rate, they are essentially uncorrelated with the folding rate constant. For the majority of the methods, the sperma correlation coefficients are small and have the wrong sign. That means that essentially if a folding event happens later, then the process is faster instead of slower. And there's only one exception, which is Raptor X, which has a correlation much better than the rest, but still not comparable to the chain length. The perhaps final experiment that I ran related to the intermediates that are detected in the pathway. I collected a bunch of proteins in my previous data set for which I had hydrogen deuterium exchange experiments that told me which pairs of secondary structural elements were formed in the intermediate and determined whether the predictions of my programs correlated with this. To make this coherent with the rest of the work, I casted the question as a classification problem, trying to identify which pairs of secondary structural elements are formed in the intermediate. And, well, the results are that actually they are all pretty bad. If you compare most of your classifiers against a random baseline, none of them is going to be superior to a random baseline, which suggests that actually these methods do not have any information whatsoever about the intermediate. In fact, I went a little bit deeper into this question and I observed I tried to explore the different intermediates. What I observed is that there is a massive variation between the predictions in independent decoys. If you write the interactions between secondary structural elements as a binary string, you label them in a simple string and assign zeros and ones depending on whether a pair of secondary structural elements is in contact or not in the intermediate. And you compute the Jacquard similarity between these binary strings, which is something that some people in the audience might know as a Tanimoto similarity from cheminformatics. What you will observe is that the majority of predictions are actually very dissimilar. If you compute the average Jacquard similarity for different proteins, as I do in this graph, the vast majority of the proteins, with perhaps exception of DMP fold for some of the factors, are on the top, on the left of the plot, showing that the vast majority of the intermediates are predicted to be dissimilar. In some of the cases, in fact, this is so important that all of the different predicted intermediates are different. There are no two identical intermediates throughout the 200 decoys. And another thing to take into account is in the cases where we have somewhat higher similarity, if you actually look at those cases in more detail, what you will observe is that there are smaller proteins that have only, say, 3 or 4 secondary structural elements, and therefore it's a lot more difficult to have dissimilarity between them. These are the results that I have obtained. So I'm just going to briefly talk about the conclusions of my work, and I'm going to try to finish a little bit early so we have time for some questions. So my first conclusion is that protein structure predictors, at least the ones that I had access to, I can't say about AlphaFold yet, are not telling us anything about the folding pathway. They are not producing the right information. They are not telling me the right mechanism. They are not producing the right folding constants, and they are definitely not producing the right intermediates. This leads me to think, also from some the correlation that I mentioned before between methods that use more physics are slightly better at predicting the folding pathway, even though perhaps the structure, leads me to conclude that the physics induced by current deep learning methods, like used in TR Rosetta, RaptorX, or DMP Fold, are unrealistic and do not seem to correlate with the experiment or anything that we're looking at. And I think the perhaps more overarching conclusion is even if we have reached a point where protein structure prediction is mostly solved, even if AlphaFold 2 and similar methods seem to have completely solved this problem, or at least for the vast majority of the cases, there is still a lot of open questions in protein folding, and there's a lot more to work on that area. I just want to finish by thanking my sponsors, EPSSC, Hoffman LaRoche, and UCB for financial support. I would like to thank my supervisor, Professor Charlotte Dean, and some of the other members of the folding subgroup in the Oxford Protein Informatics group, Dan Nisley, Ollie Kirk, Mark Janowski, and Dominic Schwartz. And of course, to all of you for your attention and for your time. And I look forward to hear any questions from you. Thank you very much.